Hello everyone, today I am going to talk on the topic High Resolution Diatom Fossil Record Reveals the Past 2 Centuries Nutrient Dynamics in Lake Tanganyika. Let me start with the introduction. Lake Tanganyika provides several kinds of ecosystem services to a large population of flora and fauna living in the lake. It is the habitat for around 250 endemic cichlid fish species and 70 non-endemic cichlid fish species. Besides that, the lake provides economic and food services to nearly around 2 million population living in the riparian zone of the lake. So the lake is directly associated with the livelihood of the people living there. So the rationale or reason for this study is we wanted to see how the diatom concentration and composition varies in the near shore region of the lake and the offshore region of the lake. So we wanted to see nutrient dynamics at different locations of the lake. Since the lake is very large, spread in very large surface area, there might be spatial heterogeneity in terms of nutrient dynamics. Besides that, we can also see the signal of climate warming in the lake. For example, due to the climate warming, the lake surface temperature increases, which causes longer stratification period and low upwelling events, which causes decline in nutrient concentration in the ephelimnion and brings decrease in diatom concentration. So we have used diatom as the indicator for this study because diatom have cell wall made up of silica, and that makes it easily preserved as fossils in the lake sediment record. Besides that, diatom are the base of the food chain, which means they are the primary producers and they supply food and energy to the higher trophic level like zooplankton and all the population of fish living in the lake. So if there is decline in diatom concentration, it is directly impacting the fish population and overall productivity of. I have already introduced that study area is Lake Tanganyika. It is the second oldest, second largest, second deepest and the longest lake in the world. It is ancient type of lake made by the tectonic activities with total catchment area of 231,000 km square, and in 2023 several cores were drilled out from the southern basin of the lake near the Sumbu wildlife area, and for this particular presentation I have only included core 6b, the near shore core, and core 11a, the offshore core, because I still need to analyze the diatom sample from the other core. So the main objective of this study is to explore the nutrient dynamics in Lake Tanganyika based on the diatom fossil records from the southern basin. Specific objectives are to identify and count diatom from each centimeter of the onshore and offshore core, and to determine diatom abundances and concentration from the near shore core 6b and offshore core 11a. So to fulfill those objectives, we collected sample in 2023 from the southern basin, and sub-samples were taken out from the each centimeter of those cores. And for diatom analysis, we followed the guidelines by Butterby et al. 2002, and we use hydrogen peroxide for digesting the organic matter. Once the sample was digested completely, it was mounted into a glass slide by help of Nafrax, and then we observed diatom with help of electronic microscope, where we counted around 300 diatom balls from each sample. If the concentration was very low, we counted at least of 50 diatom balls from those low concentrated samples. As a result, we could observe several beautiful diatoms. For example, Alucocera species, which are heavily silicified and indicates higher silica concentration in the water column, and centric planktonic diatoms, which indicate low phosphorus concentration, high phosphorus concentration in the water column, such as Cyclostephanus malavensis and Stephanodiscus mulleri. Similarly, there were long needle-shaped Nischia species, especially Nischia lacustris, which indicate low nutrient concentration in the water column because these species are adapted to the low nutrient concentration since they can absorb nutrients from large surface area, from their large body surface area. For core 6b, in this graph we can see that the green color, it indicates diatom concentration, and the other color indicates diatom genera. So from depth 27 centimeter, which dates to around 1860, diatom concentration declined and diatom concentration was higher before that. So before 1860, diatom assemblage was dominated by centric planktonic diatom Stephanodiscus mulleri and small abundances of Cyclostephanus malavensis, indicating higher phosphorus concentration in the water column. However, after 1860, diatom concentration declines. At the same time, the assemblage is mainly dominated by the thinly silicified elongated Nischia species, indicating low nutrient concentration in the water column. And from 2015, we could see increase in centric planktonic diatom, indicating rise in phosphorus concentration in the water column. And this is the result from core 11a, that is the offshore core. It shows that from before the depth 37 centimeter, diatom concentration was higher, and this 37 centimeter, it also dates around 1860. So here also diatom concentration declined from around 1860, and before that the diatom concentration was higher. But if we see the assemblage, it is a little bit different than core 6b. Before 1860, assemblage was dominated by planktonic Nischia species, as well as there were small abundances of centric planktonic diatoms. However, after 1860, in the assemblage we could see small abundances of Alocacera, indicating low silica, increase in silica concentration, and there was an interval from 25 centimeter to around 15 centimeter, which is from 2000 to 2007 CE. There was increase in Gomphonemia clavii, indicating increase in periphytic habitat, increase in lake level, and increase in surface runoff into the lake, which transported Gomphonemia clavii from the littoral region to the pelagic region. We also tried to compare the result from these two short cores with a long core taken from the central basin of Lake Tanganyika in 2017. In that core as well, we could see that the diatom concentration declined around 1860, and assemblage was mainly dominated by Nischia lacustris. However, in that core, the concentration of other benthic species was very low or even absent. So from this result we can see the spatial heterogeneity in the diatom composition at different locations of the lake. At the same time, we could see decline in diatom concentration in the recent period. So we can say that there is spatial heterogeneity in diatom composition at different location of Lake Tanganyika. There is increase in long needle-shaped planktonic diatom and centric planktonic diatoms. At the same time, diatom concentration is declining. So if we want to see healthy Lake Tanganyika, healthy fish population, and happy people living around the lake, we should implement lake conservation, sustainable lake conservation management plan. So my future plan are to complete the diatom analysis from all the 7 cores collected from the southern basin, and with that analysis and result I obtain, I will be able to describe the spatial and temporal nutrient dynamics in more detailed way. Thank you.